the recording on. There we go. Wonderful. So I'm so happy that we have Laura Reynolds presenting for our webinar today. She is an associate professor in the University of Florida IFAS Soil, Water, and Ecosystem Sciences Department. And she is a triple who. She got all three of her degrees from the University of Virginia, a BS in biology and a master's and PhD in environmental science. After which she did two postdocs, one at UC Davis and another at Rutgers. And of course, her professional interests, as many of you may know, focus a lot on seagrasses, but broadly, ecological interests in things like how diversity affects ecosystem function, all levels of diversity, whether that's species or genetic, with a strong focus on seagrass meadows. So with that, Laura, the floor is yours.
Thank you. Let me go ahead and share my screen. All right. Thank you so much. Thanks for that introduction. I have been sitting in these webinars for a couple of years every month, and this is the first time I've presented. So it's about time, and I'm excited to do so. I am going to be talking to you about one of my first loves and about one of the hallmark resources in the Nature Coast region, and that is seagrasses. The title of the talk that I'm going to give you is Seagrass Species Respond to Environmental Change Differently, Implications for Restoration Strategies. Before I start, I want to acknowledge that I have several collaborators and that have contributed significantly to the work that I'm going to talk about. And some of those folks, like Savannah and Tom Fraser and Chuck Jacoby, have worked in the Nature Coast a lot longer than I have and have taught me a ton. And some of those folks, like Alex Biak, Christine Rohal, and Jamila Roth, are members of my lab group and have done a lot of the nitty-gritty of this work. So what am I going to talk to you about? The outline of sort of what I'm going to talk to you about is I'm going to do a little bit of an intro on seagrass reproduction biology. And I know a lot of you in this room know this probably better than I do. Don't worry, that part is very short, so hopefully you won't get bored. But I do want to make sure that we're all on the same page and we don't get lost a little bit later. I'll tell you a little bit about some cool seagrass ecology, why seagrasses are important, and why we need to mitigate some of the losses of seagrasses through things like restoration. And then I'm going to talk to you about how we might use plant selection to increase restoration success when we need to mitigate for some of those losses. And then I'm going to finish up by talking a little bit about how we choose plants and how that might change when we talk about different seagrass species. So let's go ahead and get started about that little bit about seagrass biology and reproduction. Seagrasses are really cool plants. They're flowering plants that live completely submerged in salt water. Morphologically, though, they're not that different than grasses that you're probably a little bit more familiar with. Let me get a pointer going here. If you're looking at a picture of a seagrass shoot here, the sediment is going to be about right here, and this below ground structure that looks sort of like a horizontal stem is called a rhizome, and that rhizome can elongate, and once it elongates, it can produce new shoots. You see there's more of these little bundles of leaves on this one. Those new shoots are genetically identical to one another. They may be connected underground. Those rhizomes can be severed, and if those rhizomes are severed, both of those bundles of leaves can continue to live even though they are separate. Occasionally, one of these shoots will flower, and here's some pictures of some flowers. On the left, we have a flower of turtle grass. You see a little amphipod on that turtle grass flower, and we just learned in the last few years, I say the last few years, but I guess it's been a little longer now, that amphipods can actually, and invertebrates can actually help pollinate turtle grass flowers, which is pretty fun. Many of you may have seen turtle grass flowers. A lot of you probably have not seen shoal grass flowers, and that's what our picture on the right is. These flowers are very, very small. They're very close to the sediment surface. Sometimes they're even covered a little bit in sediment, and you don't see them unless you're looking for them, but they are there, I promise. Once you produce flowers, they can produce seeds. Those seeds can germinate and create new shoots. And those shoots are genetically different than the parent plants. And that's how we get genetic diversity in seagrass meadows. So you might look at a seagrass meadow and see something that looks like this. All of these little bundles of leaves that I'm calling shoots coming out of the sediment surface. You're not really sure which ones are the same clone, how many clones you have in this area, unless you bring those samples back to the lab and test them genetically. And that's what I've done here. Here, what I've done is I've laid down a 50 centimeter by 50 centimeter quadrat on this seagrass meadow, and I've mapped out these plants. Each of these dots, there's 120 dots on here, represents a seagrass shoot. And the different colors, there are 9 different colors here, represent a different seagrass clone. So there's a lot of clones in this tiny 50 centimeter by 50 centimeter area. A lot of them are this red clone, but there's several others as well. This is a 50 centimeter by 50 centimeter 50 centimeter by 50 centimeter area, but I would say that this is not atypical. This is not, if you go out to a seagrass meadow close by to wherever you are, I would expect to see something similar to this. Okay, so I promised the biology part would be quick and hopefully painless. So we're going to move on a little bit to the fun stuff. Like, why do we care about seagrasses? Not just because they're biologically cool plants, but they actually do some cool things as well. Most of you already appreciate seagrasses. That's why you come to webinars like these. And you probably appreciate seagrasses in part because of their habitat quality. About 85% of Florida's fisheries species live at least part of their lives in seagrass meadows, and a single hectare of seagrass can support 40,000 fish and up to 50 million small invertebrates. And that's pretty cool and really important. But they do some other really important things too. The biomass of seagrasses slow waves down. The below ground biomass holds the sediments in place. And what that means is it reduces sediment resuspension. So that it reduces the amount of waves, it stabilizes the sediment. The sediment doesn't go up into the water column as often, so the water is clearer and more light reaches the bottom. They also change the chemical environment. It takes a lot of nutrients to build seagrasses, and that means those nutrients are trapped in the plants, they're not in the water, and they're not fueling things like algal blooms. This modification of the environment, the energy of the environment, the chemistry of the environment creates a positive feedback. That means when seagrasses are there, they change the environment in a way that makes it a better place for seagrasses to grow. There's more light reaching the bottom, and you're going to get more plants there. Unfortunately, the negative is also true. When you lose seagrasses, you lose that modification of the environment. So you get more sediment up in the water, you get more nutrients in the water, which can fuel algal blooms. Both of those things limit light to the bottom. And when you lose seagrasses, the environment becomes a worse place for seagrasses to grow. Now, unfortunately, this decline of seagrasses, this loss of these services is happening in a lot of places, in Florida. This map is old. It's from 2016. It would probably look worse if we saw it today, but it illustrates what I want to show you, and that's that, well, first of all, each of those dots on the maps are a place where seagrasses has been declining, and the colors are the reason that it's been declining. And what I want you to get from this is that seagrass loss is pretty ubiquitous. It comes from a lot of different reasons, but a lot of those reasons have to do with reduced water quality and reduced light reaching the bottom. The other fun thing I like to point out about this picture is the area that we're interested in, the Nature Coast. There really aren't dots there, and that is one of the reasons that this place is so special. Okay, so we are losing seagrasses in a lot of different places. We're losing the functions and the services, the things that we value. And because seagrasses are so valuable, we want to mitigate for those losses. We want to restore plants. And one of the things I'm going to move to now is talking about how plant selection can help us make restoration more successful. So what plants do we want to plant in a restoration setting? First of all, we want to plant plants that are going to reproduce quickly. And the reason for that is we want to take advantage of this positive feedback. If you plant some plants, you get more plants, they're going to start modifying that environment and making it a better place. The other thing that we want to do is make sure that we're planting plants that are going to be able to withstand some disturbances because, because of this negative feedback, we don't have the ideal environment. So we need plants that are going to be resilient to small disturbances. Okay, so how do we find plants like that grow quickly and that have our desirable traits? We're going to go back to this diagram that I showed you what a normal seagrass meadow looks like. Remember, there are a lot of different clones in these meadows, and what we're thinking is that these specific clones may have desirable traits that could be related to restoration success. Okay, so the first experiment I'm going to talk to you about is just about that, about thinking about how specific clones perform differently. And this is an experiment that I did in Bodega Bay in California. This is a tank setup. In each of these tanks, there are multiple pots, and that's what this represents. Each of those colors is an individual clone. So I actually went out and cut the clones apart. So I know that, and I did verify that they are genetically identical. Then, after I set all of these experiments up, I created a stressor event. I created a warming event. So for about 5 weeks, I kept all of the tanks at the same temperature, and that's about the same temperature as the meadows nearby. Then I simulated a warming event where the water was elevated in temperature by 5 degrees for about 5 weeks. And those of you who have been around Florida in the summer recently know that that's pretty realistic. That's happening a lot. I then brought those plants back down to ambient temperature so they had a time to recover from that warming event. After each of those time periods, acclimation, that warming event, and recovery, I brought the plants back to the lab and I measured a suite of different traits: physiological traits, morphological traits, and productivity. Just for time's sake for today, I'm only going to talk about shoot production, the number of new shoots produced, and how big they were. But if you're interested in any of these other parameters, I'm happy to talk to you about them later or send you the paper that talks about this. Okay, so here is a results slide. On the x-axis, we have different genotypes. There are 7 different clones that we used in this experiment. On the y-axis, we have difference from control. And basically that means if this is at the very end of the experiment, after that, we brought everything back down to normal. If the dots or the bars were above the 0 line, that would mean that the plants that were previously warmed were doing better than the controls. That's not the case. All of these values are negative, and that's because that warming event was stressful to these plants. Importantly, though, some plants were stressed out a little more than others. You can see genotype 2, genotype 6, and genotype 7. They did really poorly. However, genotype 1, 3, 4, and 5, they did okay. They were stressed out by this warming event, but not as much as the others. So what that means is if I was going to go out and do a restoration in a place where I expected there to be warm temperatures, what I might do is I might go and go to a nursery and propagate some of these clones that are doing really well under warm conditions. But there is a caveat here. Let me take a second to explain these graphs. On the x-axis, we have initial growth, and this is under control conditions. So this is how many new shoots, we have shoot production here, that it produced during that acclimation period. So these points over here on the x-axis, they grew shoots really, really quickly. These did not grow shoots very quickly. And this is that warming response. Again, these are the ones that did well under that warming event. These are the ones that did poorly. And there's a trade-off here. Remember when we started this, we wanted plants that reproduce shoots really fast to take advantage of that positive feedback. And we wanted plants that were resilient to stressors. And this is telling us you can potentially have one, but you can't have both. And this graph on the other side is the same thing, but with how big the shoots were. And we see the same thing. There is a trade-off. Okay, so I told you about one experiment, and I want to show you that this is not an isolated thing. This idea that clones perform differently and this idea that there are trade-offs are pretty ubiquitous in the literature. This graph on the far right is the one that I just showed you from work that I did. In the middle, we have some work by Fiona Tomas, and what she did is she took clones, she named them by colors instead of numbers, but these are specific clones. And this is biomass that is consumed by, in this case, by an isopod. And what you can see here is this isopod really liked this blue genotype. So if you were going to plant into an area with a lot of isopods that might eat your plants, you're going to avoid this blue genotype. I have redone this experiment with different grazers and found that amphipods, as opposed to isopods, actually like this red genotype. So again, there is a trade-off. And this is work by Randall Hughes, and she also calls her clones by color. And this is how fast they take up nutrients. Remember, that's an important part of that positive feedback. Let's look at this orange genotype. It takes up nitrate from the water column at pretty low rates, but it takes up ammonium in the sediments at pretty high rates. Once again, these clones are different from one another. We can find a strong performer, but there are trade-offs. Sorry, let's see if I can, there we go. So that's what I just said. The take home for this is that there are best performers, but predicting the winner can be quite complicated. The next complication I want to throw at you is that traits or best performance may be controlled by things not just genetics. And let me give you a little bit of an example of that. We are all genetically different from each other, just like these different clones are. I maybe have some traits, like maybe I'm a good cook, and maybe you are good at sports. If we are selecting a team, a sports team, then you're probably the one that we're going to want to pick to put on that team, not me. But eventually, we're going to want one. Eventually we're going to want to eat, so we might want to trade that out a little bit later. However, I might get interested in sports when I'm watching you, and my traits might change, and I might become better at sports and be a better pick for that team. And that didn't happen because I changed my genetics. It happened because my traits changed without a change in genetics. And that can happen with plants too. This is an experiment where this is experimental. This is the experimental setup. This is experimental time, and this is temperature. We have some plots in black that were kept at ambient temperature the whole time. We have some that are in yellow that were kept at ambient temperature and then had a warming event. And then we have some that were primed, and that's what's in this red, where they experienced a warming event. They came back down, recovered, and experienced a second warming event along with those plants in yellow. And what they did in this experiment is they measured traits at the end. And here's what they found. I apologize if the colors don't line up with the experimental design on this. This is not my work, so I couldn't really change their colors. But this is the control treatment, the primed that had previously been warmed, and the ones that had not been primed. Those ones that had been primed at the end of that second warming event had a higher growth rate and tended to be bigger than those that had not experienced that warming event. So they didn't change their genetics during this experiment, but that previous experience with that environmental change also resulted in a change in traits and a change in desirable traits. So there's a couple of ways we can get those traits that we want. But again, that complicates things just a little bit. We can't just go to genetics. We have to think about what the plants have experienced as well. Okay, so I'm going to move on a little bit and talk about diversity. You know, we've been saying we're going to pick a high performer, but if you go to that natural seagrass meadow, you actually have a lot of different clones. And so maybe it's important to have a lot of different clones. To talk about this, I'm going to talk about some work from my dissertation. What I did was I took plants from the Chesapeake Bay and from the Virginia coastal bays. I took seeds and I planted them in an experimental design, either with each source by itself or all sources together. And over 3 years, I measured genetic diversity, plant density, canopy height, invertebrate density and diversity, and productivity. And this graph is showing us what we found for genetic diversity. Those plants from the Chesapeake Bay, from Object Bay and the York River, had a lower genetic diversity than that from South Bay and the Virginia coastal bays, and all of those seeds together. And you might be saying, Laura, that's not what you expected. You expected to put all of those sources together and get a higher diversity, and that's true. But South Bay is a restored site that was planted with seeds from all over the Chesapeake Bay, and so bringing seeds together had already been done for me. So overall, what we have is high diversity treatment and a low diversity treatment. And did that matter in terms of plant performance? In these graphs, we have high diversity on the left, low diversity on the right. We have density of plants. We have shoot height, or how big the plants are, the number of invertebrates living in those plots, and productivity. And what you see here is that in terms of a restoration setting, having a lot of different clones or having a lot of genetic diversity actually boosted the performance. So that's a second way that we might get higher restoration successes, by incorporating diversity. And the reason for this is that all of these clones have different traits. And as the environment changes, there is, if you have a lot of different clones, you have a better probability of having one that's going to do really well. And that's often called the insurance hypothesis. Okay, once again, I want to show you, I showed you some of my work, but I want to reiterate that I'm not the only one that has done this, and examples of positive effects on diversity are quite common. This graph in the middle is from the data you just saw, but it looks different. We have a measure of diversity on our x-axis, productivity on our y-axis. You can see there's a positive relationship. On the left, we have another study by Randall Hughes, where she planted 1, 2, 4, or 8 genotypes in the field. And during her experiment, she had a large grazing event where geese came and ate her experiment. And what she found was those plots that had high genetic diversity had more survival of that grazing event than plants with the low genetic diversity. This has been done enough that there are literature reviews or meta-analyses on this type of work, and that's what we have on this far right graph. This is the number of studies that have found positive effects of diversity, neutral effects, and negative effects. So really what we have here is an overwhelming evidence that, that there are positive effects of diversity. What does that mean when we're looking for plants for restoration? That means effective plant selection likely includes a balance between selecting individuals with desirable traits, either desirable traits because of genetics or because of plasticity, and maximizing diversity. Both are important, and we need to keep both of those in mind. I want to give you a little caveat about all of the work that I've just told you about. All of that work, I'm not sure why this is not showing up, but I'll explain it to you and you can still see the important part. This is a meta-analysis of all seagrass diversity studies. And on the x-axis, each of these bars represents a different seagrass species. And all of the studies that I just showed you were from Zoster marina. And you can see in the literature, this big bar is Zoster marina. So most of the studies that have been done have focused on a single seagrass species. Now this is a map of the world, obviously, and the green is the distribution of Zoster marina. Here we are in Florida. This is a temperate plant, and we do not have that plant here in Florida. So we have a question of whether this research and these findings that I just spent, a good amount of time telling you about are they relevant? Well, let's talk a little bit about why we think the effects of species might be important. Here we have different seagrass genera here, and we have their organized here by their traits. We have colonizing species over here that grow quickly, that are short-lived, that sexually reproduce a lot and have low physiological resistance. We have plants over here that are slow growing, that tend to be larger, that tend to not have a large seed bank, and that might have a high physiological resistance. Zostra, that species that is used most often, sits here pretty close to the middle. In Florida, these are the genera that we tend to see. You can see that they are distributed and that they have traits very similar to Zostra and traits that are very different than Zostra. And for the rest of this talk, I'm going to concentrate on two species that we do have in Florida, and that's Haliduli, which is similar in those traits to Zostra, and Thalassia, which is much larger, much longer lived, has a higher physiological resistance, and no seed bank. And the goal for the rest of this talk is to think mechanistically about how traits and plasticity vary with environment and how that can be different between the two species. And we're going to do this in the Nature Coast region of Florida. And that's what this map shows us. These are sites. We have Cedar Key up here and Tampa way down here, just to give us an idea of where we are. This is an interesting place because, again, it's relatively lower in stress. We don't see a lot of seagrass decline, but there is a variability in the environment. And that's what this graph shows us. We have leaf nitrogen to phosphorus ratio on our x-axis, and we have frequency on our y-axis. These sites are colored. The darker colors are these sites to the north. Lighter colors are these sites to the south. And what you see is these sites to the south have really high nitrogen to phosphorus ratios. They have really low phosphorus. And this change in environment is reflected in change in plant traits. I should note that this is from Savannah Berry. It's from her dissertation work. So fun connection there. So in this graph, it shows us how that change in environment is reflected in shoot height. This is water column phosphorus. This is shoot height. You can see if there's high phosphorus, like up here on the north, you get bigger shoots. And this is just a way to see that a little bit without all of these graphs. These are the sites to the south with the low phosphorus. These are sites to the north with the high phosphorus. And you can see these high phosphorus plants growing in a high phosphorus environment are much, much bigger. Now, I told you I was going to tell you about two species, and so far I've only told you about a single species. And that's because when I came here to work in Florida, we knew this from Savannah's dissertation about Thalassia, but we didn't really know how Haliduli varied in height across this gradient. So I went back to some data and I looked for that relationship with Haliduli, and it turns out that Haliduli has that same pattern as well. So we have all of our sites on our x-axis from our low phosphorus site to our high phosphorus site. We have plant length, Thalassia in black and Haliduli in white, and you can see that same pattern. Plants tend to get larger as you have more phosphorus in the environment. So what does that mean? It means that these populations have different traits. If we need tall plants for a restoration, we know where to go. We can go to these sites with a high phosphorus, and we can collect plants that are tall, and that might be a desirable trait for our restoration. But there's a question about whether those traits are related to genetics or plasticity, so let's take a quick look at that. Okay, so I went to most of these sites, those that are circled in red, and I took samples and brought them back to the lab, and I looked at them genetically. And the first thing we're going to ask is diversity of Thalassia different than the diversity of Haliduli? And the answer is no. These are different measures of genetic diversity: clonal richness, number of alleles per locus, expected heterozygosity. Inbreeding is not really a diversity metric, but we're throwing it in there for you anyway. where our sites going from high phosphorus to low phosphorus. And what you can see is that different sites may have different levels of diversity. These Wiki-Wachi plants have higher diversity than these Wakasasa plants, but that is true for both Thalassia and for Haloduli. Those plants tend to be behaving in the same way. So instead of just looking at genetic diversity, we're going to look at genetic differentiation. So if we have two sites, differentiation is telling us how different those two sites are. And we're going to use a metric called FST to understand that. Low numbers of FST, if an FST is 0, then you have one population. These two populations are interbreeding with one another. The biggest number you can get is 1, but that's theoretical. Most of the numbers are going to be much, much smaller than that. On the x-axis, we have Thalassia FST. On the y-axis, we have Haloduli FST. And this is just a 1-to-1 line. And what you can see is most of the points fall above this 1-to-1 line. That means if I'm looking at two sites, the Thalassia plants between those two sites are much more similar to one another than the Haloduli plants are. This is another way of looking at that. What I did is I took all of those samples and I fed them into a model. That model told me that for Thalassia, there are 2 genetic clusters on this region. And the pie graph shows us the proportion of samples that were assigned to each of those genetic clusters. And you can see these are all about 50-50. Importantly, this model will never tell me that there's one genetic cluster, but what this is telling me is all those populations look pretty darn similar. Conversely, for Haloduli, what you can see is the model told me there were 3 genetic clusters, and each of these populations is dominated by a different genetic cluster. That means they're genetically distinct from one another. This is not particularly surprising if you know about the biology of these plants. Thalassia has fruits that are positively buoyant. They float, and they can float for many, many miles. These sites are about 50 kilometers apart, or from the northernmost to the southernmost is about 50 kilometers, and fruits can definitely float that distance. So it's not surprising that they are exchanging genetic material. Haloduli, remember, those flowers are very close to the sediment surface. They're very small. Sometimes they're covered by sediment, and the seeds are negatively buoyant. That means they probably don't move very far. There are other ways that you can get genetic connectivity through floating fragments or birds moving seeds around, but it's not surprising that we're having less connectivity with Haloduli. What does this mean, though? It suggests to me that this difference in plant height might be genetic for Haloduli, but might be plastic for Thalassia. To test this, we did a little common garden experiment where we grew plants from each of these locations in tanks at the Nature Coast Biological Station. I know that these are not very pretty pictures. I did this experiment with a postdoc at the time in my lab group, Christine Rohal, and this was a year-long experiment, and we both had babies during this experiment. So a few things may have slipped through the cracks, like taking pictures, but I promise you the data is legit and well taken care of. And here are those data. We have time in months on our x-axis and height of plants on our y-axis. We have Thalassia here, as expected when we collected these and we planted them. These Haloduli plants were tiny. At the end of the experiment, those plants became very, very similar to one another. In fact, they were not statistically different from one another at the end of the experiment. So this suggests that these are plastic, that they're changing their traits with their environment. Haloduli, on the other hand, started out different from one another, and at the end, these plants from Crystal River that were tall initially stayed tall at the end. And that suggests that this height is potentially driven by genetics and not just plasticity. Okay, so what does all of this mean? These two species have similar morphological response to changing environments. But Haloduli, we think, is that response is genetic. It's local adaptation to conditions where that it has been experiencing. These populations are heterogeneous, so populations that are not very far apart from each other can be quite different, especially if their environmental conditions are different. Thalassia is just the opposite. These populations are homogeneous. It looks like they're a single interbreeding population, and we're getting this morphological response because of plasticity. I said I was going to think about diversity and traits. Importantly, diversity is the same between these two species. This has implications for management in a changing environment. What we might think is that if the environment changes, that Thalassia will be able to change its traits to survive and even thrive in this new environment. Or Haliduli might not. It also has potential implications for the importance of biodiversity ecosystem function relationships. If these plants can change their traits, then having a lot of different clones may be less important for a species like Thalassia. I just added in here that the genetic diversity is the same. Okay, so what does this mean in terms of plant selection? I said at the beginning of this that effective plant selection likely includes a balance between selecting individuals with desirable traits, either through genetics or plasticity, and maximizing diversity. I think that is true for both Thalassia and for Haliduli. If you haven't noticed, for the last couple of slides, I have said may probably a lot of different times. And the reason I'm so cautious when I'm talking about this is that this study has a relatively limited spatial scope in an area where there's not a lot of stresses. We used neutral, less powerful genetic markers, and we only looked at really a single trait, and that is plant height. And in order to be more confident in these findings, we need to repeat these experiments in different places. There are also a lot of managers on this call, and I can hear the groans right now, even though everyone is muted like they're supposed to be muted, and that how are you going to select plants when I'm giving you this sort of wishy-washy statement of how you need to balance these things. You don't even know which populations are different from one another. And I hear you loud and clear, and that is something that I am working on and hoping to come up with better tools that we can use to implement these ideas. And one of those tools is working with the Seagrass initiative with Moat Marine Lab. This is a legislative funding project. And one of the things that we're doing is we are going to map seagrass genetic diversity over the state. So we're going to look beyond just the Nature Coast region, and these dots that we have here are those studies that I just showed you. You can see we have a lot of dots already, and we're going to spend the next year filling in those dots. So I can tell you whether this homogeneity of Thalassia is a Nature Coast thing or whether it's a statewide thing. I could tell you whether this heterogeneity in Haliduli is, again, just a Nature Coast thing or a statewide thing. And hopefully we'll be able to use tools like this to better inform management. The other thing that we're going to do with this is we're going to stockpile a lot of DNA from seagrass meadows. And once we are ready to use more powerful genetic markers, we're going to have that DNA in reserve so we can answer those questions quickly and hopefully package them in a way that can be used effectively by management. And with that, I will stop talking. I'm happy to answer some questions, but my contact information is up here. And if I don't get to your question, or if you want to ask it in private, then please reach out to me, and I'd be happy to answer those questions.
Awesome. So yeah, feel free to put your questions in the chat, everybody, or unmute as well.
I'm going to stop sharing so I can see the chat.
Okay, we just had one come in.
All right.
Yep. So the first one, do Thalassia and Haliduli have different rates of sexual reproduction in the study region? And does Thalassia rely more on clonal growth than Haliduli?
That is a great question, and one that I cannot effectively answer right now, but it is one that we are working on as part of our monitoring projects in the Nature Coast region. We will have an idea of how often Thalassia flowers. It's harder to know that about Haliduli, but we are looking for seeds in each of these regions that will give us an idea behind that. The fact that our clonal diversity is different between different sites, but it's low for Haliduli and Thalassia, it's low in the same places and it's high in the same places, suggests that they have similar trends. But again, that's a suggestion and something that we should be looking into more carefully.
Awesome. Next question is, how did the grasses shrink in the mesocosm experiment?
Yeah, good question. This was a year-long experiment, so leaves sloughed off. In many cases, we had new shoots produced, but in this amount of time, all of the leaves would have turned over. So that's how they shrank.
Cool. Are you interested in other populations for your future studies beyond those that you showed on the map?
Yes, thank you for asking that. I should have said that. Yes, we will be filling in that map a bit over the next year and adding more dots. That is absolutely the goal of that study is to fill that in much better. Those dots are spots that just through my time at UF that I have samples in my freezer that not necessarily analyzed together yet, but they will be analyzed together, and we will be filling in and adding many more dots to that map. But thank you for asking that question, because I should have said that more clearly.
I have a question that
Yeah.
I think I know the answer to, but other people on the call might be wondering, what about Serengodium?
Yeah, good question. Um, you have to start somewhere. And I would say that right now in Florida, Haliduli and Thalassia are the species that we are concentrating on the most. There are people on this call and people in Florida who are pushing us to think more about Serengodium, and we absolutely should be doing that. And that is in the long-term plan.
Cool. Are you considering looking at populations of Thalassia in Mexican waters?
For this project, not necessarily, but it is absolutely something that we know that these fruits can float long distances, and that you know plants don't care about international borders. So yes, that is a very relevant thing right this second. I don't have funding to go do that, but looking across a broader geographical range is a very logical next step. There are some papers who have compared Thalassia genetics across the Caribbean. They happened in that study, again, because you can't get everything, had missed a lot of the Nature Coast region, but we do have some background information on Thalassia from Mexican waters as well. But putting that together in a large picture absolutely is something we should be thinking about.
All right. What do you see as the knowledge gap which is having the greatest impact on seagrass restoration at this time?
Yeah, that's a good question. And I think there are a lot of knowledge gaps. I think in terms of practitioners, the question that I get a lot is how far we should be moving plants, and should we be growing super clones, or should we be looking at high genetic diversity? And that's really the motivation behind the work that we're doing with the Seagrass Initiative and Moat Marine Lab. But there are a lot of other knowledge gaps as well. There's a big problem of how do we protect seagrasses from herbivory? If all the seagrasses are gone, all those things that used to love to eat seagrasses are going to chomp on them as soon as you get a few planted. So I think the lessons are very site specific, and I think that I'm working on this one because I think it's a very important data gap, but it is certainly not the only one.
Excellent. So you touched on this a little bit, but there's a question basically saying, enjoyed the example of showing where you can find taller plants, and with these areas being relatively close together, are there any concerns for transplanting, like introducing, I guess, different clones to different areas, et cetera?
Yeah, absolutely. That's a good question. And if you are moving plants around, there's always potential for moving things like pathogens around. So you need to be careful about that. This work would suggest, in terms of local adaptation, that moving Thalassia around this region would not be a big deal because those populations are genetically similar. For Heliduli, it might be more of an issue. But again, that is very preliminary, and I'm adding all of those mays in that I added at the end of my talk, because that's what I think is going on. But in order to say that in a way that might affect where we move plants, I would want to do some more studies.
All right. And do you look at any other water quality variables in addition to phosphorus? And was there a measure of turbidity during the recent study on phosphorus?
Good question. These sites were chosen based by Savannah, to be honest. As part of her dissertation, is a lot of water quality monitoring that goes on, and these sites were chosen to be very similar to each other in everything except for phosphorus. And the things that we measure are salinity, water depth. We don't necessarily measure turbidity, but we do measure light attenuation. And again, these sites were selected because they were very similar in everything except for phosphorus. But all of those other things could absolutely affect plant traits and should be investigated. Again, that's one of the reasons that the study is limited. We're looking at the variation in one environmental parameter, one trait, and as we talked about at the beginning, there's lots of trade-offs, and things need to be, all of these things should be included for a full understanding.
Awesome. Well, thank you so much to everybody that submitted questions. Super engaged group this time. We really appreciate you coming and joining us on Nature Coast Currents. And our next talk is coming up on October 16th, also at 2 p.m. on a Wednesday. And we're going to have a really cool talk that many of you probably haven't heard about before. And it's ways that the algae gracilaria might be able to be used as an additive in cattle feed. And we think this is just such a cool thing. So we hope that you will join us, and I will send out the registration link for that as a follow-up when the recording for this webinar is available. So thank you all so much, and we hope to see you back next month.
Thanks for coming, you all, and thanks for the great conversation.